Welcome back to Acing AP Biology. After introducing DNA replication in the last lesson, we will be going over the process of DNA replication in detail in this video. Recall that DNA only grows from the 5' end to the 3' end, and that replication always occurs towards the replication fork. Therefore, since both strands in the parent DNA molecule are antiparallel to each other, both daughter strands cannot be synthesized continuously. For the leading strand, the 5' to 3' direction is towards the replication fork, and so it can be produced continuously as a single polymer. The leading strand only requires one primer. The lagging strand is produced in fragments because the 5' to 3' direction for this strand moves away from the replication fork. The series of short fragments that make up this strand are called Okazaki fragments, each produced in the 5' to 3' direction. As more and more fragments are produced, the bubble continuously opens up. DNA ligase, an enzyme, glues fragments together by catalyzing the formation of covalent bonds between fragments. The lagging strand requires several primers. Now let's go over the actual process. This is the 5' to 3' direction for this strand, and it is the opposite for the other parent strand because the strands are antiparallel. This is the replication fork. Helicase untwists the two strands, and the single-stranded binding proteins keep the strands apart. Let's start with the leading strand. As you can see, its 5' to 3' direction is towards the replication fork, because the new strand that forms is antiparallel to the template strand. After the primase protein lays down the first RNA segment, DNA polymerase III attaches and begins adding DNA nucleotides in the 5' to 3' direction. In this manner, the leading strand is produced continuously in a single polymer. As the strand continues to grow, DNA polymerase I, another form of DNA polymerase, replaces the RNA primer with DNA nucleotides. For the lagging strand, the 5' to 3' direction is opposite of the replication fork, so the strand has to be produced in fragments called Okazaki fragments. Primase lays down the RNA primer for each fragment. DNA polymerase III attaches and adds nucleotides until the next primer is reached. Afterwards, DNA polymerase I replaces the RNA primer with DNA nucleotides. Finally, DNA ligase glues the fragments together to form a continuous strand. As both strands elongate, the bubble continues to grow in the direction of the replication fork. Recall that there are several replication bubbles across the DNA molecule, each with two replication forks going in opposite directions. Eventually, the bubbles merge and the entire DNA molecule is replicated. Now let's go over the Okazaki fragment process. First, primase lays down RNA nucleotides, serving as the primer. This is the origin of replication for the leading strand. Remember that the replication bubble goes in both directions with two replication forks, so this leading strand is for the other replication fork, which was not included in the last diagram. In step 2, DNA polymerase III adds DNA nucleotides to the primer until the next primer is reached, which in this case is the primer for the leading strand. After reaching the primer, DNA polymerase III detaches, and this is the first Okazaki fragment. In the meantime, Primase has laid down other primers for more Okazaki fragments. DNA polymerase III can then begin synthesizing the second Okazaki fragment and attaches once it reaches the primer from the first Okazaki fragment. As the lagging strand continues to be produced, DNA polymerase I replaces the RNA primers with DNA nucleotides. As the bubble continues to grow at the fork, primers are added continuously. DNA ligase glues the fragments together in this region, and the lagging strand in this region is then complete. DNA replication is a very efficient process, but mistakes do occur. Fortunately, there are several mechanisms in place to fix errors. Two major repair procedures are the mismatch repair and the nucleotide excision repair mechanisms. In mismatch repair, mistakes are corrected while DNA is being produced. DNA polymerase proofreads DNA by removing incorrectly paired nucleotides and replacing them with the correct nucleotide sequences. The nucleotide excision repair process occurs in existing DNA, where accidental mutations in DNA are corrected. These mutations can be caused by exposure to reactive chemicals. Enzyme nuclease is in charge of this process. Thymine dimers are generated by exposure to UV light like sunlight, which is why nucleotide excision repair occurs often in skin cells. This is a thymine dimer. Nuclease cuts the DNA at 2 points to remove the mutated DNA. Then, DNA polymerase places the correct nucleotides using this DNA strand as a template. DNA ligase then glues the new section of DNA to the original strand, sealing it completely. DNA ligase then glues the new section of DNA to the original strand, sealing it completely. That's it for DNA replication. In the next lesson, we will go over the process of DNA packaging into chromosomes. See you then. For more bio content, hit subscribe.